The overall goal of this procedure is to demonstrate a method for growing an inducible flax variety under conditions that result in heritable genomic variation. Furthermore, genomic changes are monitored, and the growth of the induced plants in subsequent generations is observed under the various environments. To accomplish this, flax is grown under a variety of inducing conditions. Leaf and meristem tissue is then collected at various times during the growth of the plants under the various environments. Next, DNA and RNA are isolated from the tissue samples. The final step of the procedure is to perform PCR using the DNA samples and qRT-PCR using the isolated RNA to identify genomic changes and gene expression changes resulting from growth under different environmental conditions. Ultimately, results can be obtained to show the genomic response to growth conditions with the appearance of the LAS1 insertion element at a specific point in the genome and expression differences associated with the presence of that element as visualized by PCR and RT-PCR performed with nucleic acid isolated from the tissues of treated plants.
Hi, I'm Cullis from the Department of Biology at Case Western Reserve University.
I'm Cory Johnson from the Cullis lab.
And I'm Tiffanie Moss, also from the Cullis lab.
And I'm Lukacs, an undergraduate in the summer program for undergraduate research at Cullis lab.
We use this procedure in our laboratory to study changes in the genome and gene expression resulting from variations in the growth environment.
So let's get started.
To grow flax under inducing conditions, begin by filling 5-inch pots with soil and firming. Next, plant 3 flax seeds per pot 1/4 inch deep, then firm the soil over the seeds. For the non-inducing control conditions, water the plants with 100